everybody. I am Alan Russell. Those letters there are CSO, is Chief Scientific Officer of Edgewise Therapeutics. And I am joined by Joanne Donovan. Joanne, do you want to introduce yourself?
Hi there. Nice to speak to you all today. I am Chief Medical Officer at Edgewise Therapeutics, and thank you for joining.
And we would be joined by Abby Bronson, but I think she has been defeated by the online Zoom system we have here. So hopefully she'll be able to join in a bit. But otherwise, we have us shiny two faces to introduce this program. And we'll take you through essentially some details about the program that we have here at Edgewise Therapeutics and why it might make a difference in the context of Duchenne and Becker muscular dystrophy. Okay, hopefully you guys can see the slides, and I will see if I can advance them. Which of course I can't. There we go. All right. Now we are a public company. And that means I have to show you this kind of disclaimer to just under a regulatory context, and it's really to make one point, and that of course is that our lead compound EDG-5506 is at this point an investigational drug and is not approved anywhere, right? So we are still in investigational mode with this compound and have no clinical approval as yet. So Abby would be introducing these slides, but lucky you guys, you have me instead. And then I will move on to some biology, just showing an overview of what does dystrophin do and how are we affecting the system. And then Joanne will take it over to tell you about what we know about the compound in the clinic so far. So Edgewise Therapeutics was founded in 2017. I was one of 3 co-founders of that company. Just a bit of background on myself. I've been working in muscle physiology for almost 20 years now. I actually trained as a pharmacist and did a postdoc in the postdoctoral position, that's the technical term, in the United States before joining biotech. And accidentally fell into the world of muscle physiology, and I've just stayed there ever since, and now have somewhat of a reputation, unfortunately, in that area. What I find fascinating about the area is that while it is an enormous organ, it takes up about 40% of our lean mass. Very little is understood in terms of how we might change muscle for therapeutic gain. Lots of people have tried and failed. And certainly over my 20 years of experience, I've seen a lot of water under the bridge. And in many ways, that helped inform how we went forward with Edgewise and the kind of strategies that we might employ to improve the lives of folks with rare muscle diseases, with the centerpiece of Becker and Duchenne muscular dystrophy. We want to make your lives better. That's the goal. That's the thing that motivates us every day. It motivates me. You know, these myopathies are grave and they need real therapies. Now, our strategy is what they call a small molecule strategy. So regular medicines. We do not have a genetic approach. So our real goal is to devise a therapy that's appropriate for a broad section of the population. We really want to find a real-world therapy for folks where everyone can take it and benefit from it. The lead program, as I mentioned, is this thing, EDG-5506, focused on Becker and Duchenne muscular dystrophies. And the status, Joanne will give you more of an update on that. We're currently in phase 2. Okay, so we've gone through the healthy volunteer kind of proof of concept stage, and now we're going into larger studies with both Becker and Duchenne muscular dystrophy individuals to see what it does. Now, this is Abby's slide, but I will put my patient advocacy hat on, if you like. A centerpiece for us is you guys, is the community, because we're in rare disease. We're not in large population types of conditions, right? This is a much more personal endeavor. We need to hear from you. You need to hear from us, right? And our goals here, as bulleted out, is we need input into how our clinical trials are designed, right? We want to run the best studies that we possibly can. We want to contribute to understanding the therapeutic context for risk and benefit. And we want to work with advocacy organizations to align community needs and shared goals and serve as a bridge to connect the research ecosystem to find new and better treatments. We want to provide you resources to create disease awareness and help educate individuals and families, right? That being a public company, there's only so much you can say at any one time, but we really want to supply you the best information that we can and design the best studies that we can. Okay, this is now me transitioning to me. Okay, so the first thing I want to describe is whereabouts in muscle dystrophin is. You've probably seen thousands of these presentations, and it's usually one slide with this big complex molecule in the middle and lots of stuff around it. I'm going to hopefully flesh out a little bit for you whereabouts the protein is and what it's doing. So let me turn my laser pointer on. What this is a large muscle attached to bone. So this is a tendon here attached to the bone. And what we've done is we've chopped the muscle straight down the middle so you can see all of the little fibers. Each one of these is an individual muscle fiber. We've broken one out here. Now, dystrophin is essentially lying on the outside of these muscle fibers. And it spans, or the complex that it's associated with, spans the membrane. So this is the sarcoglycan complex. It's a load of proteins. And dystrophin is this long linker protein that sits on the inside of the muscle. Okay, so on the outside of every muscle fiber is a decoration of these complexes with this thing attached to it. And then on the outside of the muscle, there's these laminin molecules here which connect the whole thing together. So essentially what you're looking at is a bridge between the inside of the muscle fiber and the outside through the membrane. And dystrophin plays this important role to attach the inside of the muscle to the outside. Now, our muscle fibers are made of slow and fast fibers to support all levels of activity. You may have heard those terms before, slow and fast. I often use the analogy that this is kind of light meat and dark meat on a chicken, right? So dark meat, those are your slow fibers. They tend to be active more often and they're more resilient. And then the fast fibers, so these are the light fibers, the white meat on the chicken, they tend to be less active more frequently, but then they control power maneuvers. And the combined activity of slow and fast helps you to support all levels of physical activity. Dystrophin is present in every one of those muscle fibers, decorating the outside. Okay, and an important note here is fast skeletal muscle fibers are more prone to injury. That's the case in healthy individuals, but also people with Becker and Duchenne muscular dystrophy, where researchers show that those fibers, these fast ones, turn over at a much greater frequency than the slow ones. Now, as I mentioned earlier on, we do not have a genetic strategy. We have a small molecule strategy designed to stabilize muscle. And that gives us certain luxurious positions. And one of those is that we can view both Becker and Duchenne as a continuum of severity. So rather than individual conditions, one marked by truncation of the protein and one marked by absence of the protein, we can see them as a continuum. Our therapy is designed to protect muscle independent of your dystrophin status. So instead of these two great buckets of Becker and Duchenne, what you have instead is a sliding scale here of how much functional dystrophin you have, from absolutely nothing to normal levels, and then a sliding scale of physical challenges from ambulatory difficulties to late onset wheelchair to early onset ambulatory problems. Okay, so that gives us this kind of unique position where we can treat both Becker and Duchenne, and we can also treat those individuals who kind of fall between the cracks, if you like. Folks with in-frame deletions, so they have a truncated dystrophin but very severe symptoms. So they look more like Duchenne, but they're diagnosed as Becker, and vice versa. Now I'm going to return now to a more complicated figure, but has the same elements. So again, this is the muscle membrane, just the same as I showed you on the previous picture. And this is the dystroglycan complex. So this is the thing that goes through the membrane and attaches to the matrix outside. And here's dystrophin again, this orange chain of proteins. Okay. Now the function of dystrophin, as I mentioned, is to span the membrane and connect the inside to the outside of the muscle. And it also connects fibers to fibers. So here's another muscle fiber sitting right next to this one here. So that when this fiber contracts, this one also gets pulled along for the ride, okay, by these connections here. And the reason that that happens is so that the stress of muscle contraction can be spread across the muscle, avoiding excessive stress on individual muscle fibers. Now, muscle fibers are designed to contract. That's what they do. That's how they bring us the functionality that allows us to get around and move, right? That movement is initiated through two proteins. The first protein is this big blue rod here. That's myosin. And here's the other one, the green filaments, and those are called actin. Now, myosin attaching to actin is the thing that causes the force and the contraction and the stress in muscle, okay? So when myosin and actin interact, the muscle contracts, dystrophin allows the load to be spread between and across muscle fibers, and that allows you to function in a normal fashion. If dystrophin is not present, so this is the kind of the Duchenne example. If it was the Becker example, dystrophin would be present, but it would be shortened in some way or destabilized. If dystrophin is not present in this example, what happens is when the myosin and the actin interact, when the stress starts in the muscle, it's more focused towards individual fibers. There's no sharing the load between muscle fibers. And that causes an excessive stress response, and it triggers well-defined pathways that cause the breakdown of muscle. And one of those pathways is membrane stress. So the muscle contracts and it transmits a stress response to the membrane, and that triggers these collapse phenotypes. So you get this excessive stress, and that causes breakdown of the muscle fibers. Okay? That's where the CK comes from, for instance, and we'll be talking about that in the later slides. So when these muscle fibers undergo the excessive stress response because they can't transmit the load, what happens is they break down and they have to use inflammation and stem cell regeneration to grow back new muscle fibers. And that happens, but of course, as the process goes on and inflammation gets larger and age gets higher, what tends to happen is that regeneration slows down, and then that leads to fibrosis, so replacement of those regenerating muscle fibers with collagen and other types of connective tissue and fat, and that's really what leads to the functional decline. So the excessive stress causes a breakdown, needs regeneration, and regeneration slows, and then you end up with the fibrosis. So our strategy is to go to the source of the stress, and that's this myosin-actin interaction. So what we found is a strategy to stabilize this myosin so that when muscle contracts, it now no longer stresses the membrane. Okay, so we haven't replaced the dystrophin. What we've done is we've just tuned the engine so that the engine can run without causing the stress response. It's a completely different strategy that has not been attempted to date in any dystrophy. And it's more of a structural approach than a genetic approach. And that's why I can kind of talk about looking at Becker and Duchenne with the same lens, right? Because it doesn't matter what kind of dystrophin you have here. you're stabilizing this part of the motor. So just to recap, and then I'll show you a little bit of biology. As I mentioned, fast muscle fibers are more susceptible to damage due to the lack of functional dystrophin. We've made an investigational therapy, which is called EDG-5506, that is designed to protect fast muscle fibers from damage by modulating myosin. So we selectively modulate those very susceptible fast fibers. In diseased animal models, 5506 protected susceptible muscle fibers and prevented long-term development of damage. So essentially, when you give the compound, it protects those muscle fibers so they don't break down, and that gives you short and long-term benefit. Here's an example of the compound in action. So what you're looking at here are two muscles from the dystrophic mouse. It's called the MDX mouse, and they're really tiny muscles, and they've been cut out and put into a little dish. This one has been treated with nothing, and then this one we treated for an hour with this compound 5506. We gave the muscles a small concentration of the compound, just enough to modulate the myosin so that the stress response didn't occur. So what you're looking at on the left-hand side as these muscles contract now, you see the muscle is moving outside of the twitch, which is the very sharp thing. And the reason for that is the membrane is becoming upset, and that's causing these hypercontraction stress phenotypes. You can see now that that's turning on proteolytic enzymes. So these are proteins that chop the muscle up and allow it to be disposed of. And you can see these things just retracting now. It's almost like you took a rubber band and you cut it in the middle, and they're pulling away from each other. And you can see as these muscle fibers scrunch up, they turn into these kind of knots here, and you see increasing numbers of these knots appearing. I haven't talked about the right-hand side because it's not that exciting, although it is extremely exciting in terms of a therapy. So this thing is just steaming along. It's contracting, but there's no stress response now, right? You don't see the appearance of these knots. You don't see any kind of movement when the muscle shouldn't be moving. So even though it doesn't have the dystrophin protein, what we've done is we've normalized the contraction process so that the stress response now doesn't happen. So the motor is carrying on as it was before, but now not leading to the destruction. So if we could achieve this in human muscle, that would potentially have a profound impact on disease progression. The last piece of preclinical data I have, so this is the animal data that we've done, is in dogs. So these are the golden retrievers with muscular dystrophy. This is the most severe muscular dystrophy model that exists in large mammals, and so is closest, if you like, to us humans. And what we did is we did two short-term studies in older dogs. So these were dogs that are highly symptomatic, so they're not pre-symptomatic. They're kind of akin to older type Duchenne individuals, something around 10 plus. And we looked at two things. We did 2-week dosing in these dogs, and we looked both at plasma creatine kinase. So that's one of these proteins that leaks from muscle. And Joanne will be telling you about that later. So this is a biomarker of muscle injury. And then in a separate study, we gave the same dogs dosing and looked at how active they were. So if you looked at that video that I showed you before, the theory would be if we give the compound, then the muscles should be able to contract normally, but not now leak and show signs of injury. And that's what we saw. When we dosed the dogs with 5506, we saw a rapid drop in creatine kinase of about 60% over the dosing period, and actually that reversed when we took the compound away. At the same time, when we repeated the study with the same dose, we saw a 30% increase in activity. So not only were their muscles becoming less injured, the dogs were actually more active during the dosing period. They were resting less and they were active more, so their muscles presumably were feeling more healthy. So a really positive sign, I think, that the compound is doing what we think it's doing, stabilizing the muscle so that it can be functional without injuring itself. Okay, I'm going to pass the baton now to Joanne, and she'll tell you how far we've progressed in the clinic and the kind of data that we've got so far.
Great. Thanks, Alan. So, as Alan told you, when muscles are injured, they release these muscle protein biomarkers. And there's a number of different ones, including creatine kinase, CK, that you've all heard about. And there's others like myoglobin. There is fast troponin, which is very specific to the type of muscle that is protected by the muscle fiber that's protected by EDG 5506, and a number of other muscle proteins. And we can measure those because we can draw blood during a clinical trial and we can measure those. On the next slide, you know that CK is very elevated in Duchenne. It is less elevated, but it is still elevated in Becker muscular dystrophy compared to control or unaffected folks. Similarly, fast troponin, which comes from these fast muscle fibers, is quite elevated in Duchenne, elevated in Becker, and we don't see it at all in the control population. It's only expressed in the fast skeletal muscle fibers. So what we do in phase 1 is we look at some of these things in order to get an indication of whether or not the drug is doing what it's supposed to be doing. So we did a phase 1 study in unaffected adults first. We look at single doses, then we look at 2 weeks of dosing. And what that tells us is, first of all, what do we know about side effects? We dosed EDG 5506 at multiple different dose levels for 2 weeks, and we saw that it was well tolerated. We didn't see safety concerns in the kidney or the liver. We saw the lab tests were normal. And what we then did was then enrolled several patients, 7 patients with Becker muscular dystrophy. And I have to give these guys absolutely a gold star, because in this clinical trial, we actually asked them to come into the clinic and stay there for 2 weeks. They were there for 2.5 weeks. So really significant contribution to the research, and we're very grateful to them for that. And the side effects were similar in unaffected adults as well as those in Becker muscular dystrophy. The most common thing was dizziness and sometimes tiredness. All of those were mild and they went away on their own. What we also learn is from this kind of a study is how best to give the drug. Now, EDG 5506 is orally available. We found that it was well absorbed. It can be taken once a day, and it can be taken with or without food. So that makes it more convenient. Probably the most important thing to find out, which we did with muscle biopsies, is does it get to the muscle? I mean, that is a key in looking at any drug, is getting to where it's supposed to be. And we found that EDG 5506 is quite concentrated in muscle compared to the bloodstream. And that tells us that it is specifically getting to where it needs to be to protect the muscle. Now, the other thing we did in the participants with Becker was to look at the biomarkers. Now, they were treated for 2 weeks. That's not very long in the global scheme of things. But what we saw was, compared with the unaffected folks with Becker had elevated CK levels. When they were in the unit for 2 weeks, they are less active, and their CK levels went down. You probably know that when you are very active, stress your muscles, then the CK goes up. When you aren't very active, like you're in a unit like this, they go down some. With Edge 5506, we saw the CK go down about 70%. So very close to the normal, to the levels. We also looked at fast troponin, which is specific to the fast muscle fibers that Edge 5506 is affecting. And before treatment, it was elevated. The participants that got placebo changed minimally, and the participants that received Edge 5506 had almost normalization of the fast troponin. And we saw this across a number of different biomarkers. We'll be presenting some of this at the MDA meeting later next month. And so where that leads us to is that gives us the information to plan additional studies. First of all, we have an open label study that has enrolled the participants with Becker who were in the first study. So those folks who were there for 2 weeks got the opportunity to continue on. And so that trial is ongoing, and it's you can find out more about it at clinicaltrials.gov. We are also doing a natural history study. There is never enough known to understand the disease. So we are working with a number of expert centers to look at the natural history of the disease. And that has actually begun. We are also planning a study in adolescents and adults with Becker who are ambulatory, and that will be starting at a number of centers in the United States in the first half of this year. So we don't have a trial number on clinicaltrials.gov, but stay tuned on that. And we are also planning an initial study in boys with Duchenne in the second half of 2022. So as Alan said, we believe that this mechanism would be effective not just in the complete absence of dystrophin, but also in the partial absence, because the same problem is there, that the stress to the muscle ultimately damages the muscle. So to kind of summarize, this is an oral therapy. We're developing EDG 5506 for the treatment of both Becker and Duchenne muscular dystrophy to preserve and potentially improve function. We believe that will happen with extended dosing in patients with any mutation by preventing damage to muscle. It has the potential to be used in combination with other therapeutic approaches, with existing treatments, as well as others in development. And the idea is we're really trying to stop the damage at the very first step, where the mechanical stress on the muscle causes damage. And this is it's kind of an edgy way of doing things, a novel mechanism that really hasn't been approached before. And we're very encouraged with what we've seen, even in the first patients that were dosed, to see those kind of biomarker responses. And that will help guide us. We'll be measuring that in the future trials as well as functional measures. So this will be available later recording, but you can visit our website to find out more, to let us know. And with that, we can open it for questions. And
Joanne, it might be worth pointing out that Abby has joined us, and you folks who are contacting that info@edgewisetx.com, you'll probably end up in Abby land. So Abby, you want to just say hi? Oh, you're muted.
I'm so sorry. For some reason, my PC was upgraded recently. I don't think it was letting me download this program very easily. But anyhow, I'm so glad to be here and so glad you all joined. And thanks a lot to MDA for organizing this, because we couldn't reach you and talk to you if it weren't for the MDA and everything that they're doing in the community. So thanks. We have some questions? At least I can field some questions for you guys. So first is one about the biomarkers dropping, the CK dropping. And I don't know, Alan or Joanne, it says, do we know how much decrease in CK was due to inactivity or was it due to treatment? Can we separate that out? I don't know which of you wants to field that one.
Mm-hmm. Yeah, so it does vary. I think what impressed us was that all of the biomarkers moved in the same direction and were basically very close to normal. That's something that you don't see spontaneously. So it is very encouraging. Of course, it's not the last word. That's why we do need to move on to phase 2 studies and understand better the effects of the drug.
Alan, this one's for you. So what about the heart? We know that in Duchenne and Becker, there's a lot of concern over heart, and it might be different for each disease, but do we know anything about the impact of EDGE 5506 on the heart?
Yeah, that's a great question. So just a reminder, the approach that we have selectively targets these fast skeletal muscle fibers, which we think are the most susceptible ones. And we're made up of about 50% fast and 50% slow fibers. Now, of course, the fibers in heart are more like the slow fibers. We don't directly target the heart at all. We're going to affect the peripheral muscles, if you like, the skeletal muscles. We're not going to directly affect the heart. Now, saying that, in preclinical studies that we've done in the DBA2J MDX mouse, that's a very long word to say, it's a severe Duchenne model in a mouse, and they get nasty heart problems. We actually saw quite nice cardiac improvements too, right? Now, that doesn't mean you're going to see them in a human, but certainly I'd rather see that preclinically than not. And what that, I think, tells us is because we're improving whole muscle health, skeletal muscle health, there's a knock-on to the heart. So even though we don't directly affect the heart, we'll be looking at cardiac health because we might see a benefit there too.
We have a couple questions about functional improvement measured. Do we see any improvement in the North Star? Have we seen any functional changes in patients to date? And Joanne, you can answer that, but yeah.
So this in this trial over 2 weeks, we didn't expect to see functional improvement. One thing we did measure over the course of the trial was muscle strength. And we saw that even in patients who had the least strength, they were stable over time. We didn't expect it to change, to improve over just 2 weeks of treatment. The muscle needs time to remodel and improve. In future studies, we will be looking at that, though. We'll be using the North Star as well as other measures to follow function over time.
Another one about sort of trial design is in the future studies that you're talking about, Joanne, do the boys have to be on a stable course of corticosteroids to participate?
So we think that this can add to the standard of care. It can be on top of corticosteroids. And additionally, potentially has the chance to be used with other therapies, with exon skipping therapies and other therapies that are in development. Because I think that the experts are kind of the opinion that this is not going to be something that's solved with one approach. We need more than one.
And for Becker?
And for Becker, they do not need to be on corticosteroids. Generally, folks with Becker are not on steroids.
We have a question. Actually, Alan, you might be able to answer this one. It's just something to ponder. So, are doing any workouts recommended when you're taking this drug? And if so, what kind of workout would they be? More cardio, more strength? Do we know the answer to that?
That's a jolly good. That's a really good question. Okay, so I'm going to give you a hypothetical answer because, of course, we don't know the true response to the compound yet, right? But let's just imagine it's doing what we think it's going to do based on our preclinical studies. And that is when you take the compound, it should stabilize your muscle so that when you use your muscles, they now no longer get injured, right? And that should theoretically allow them to have a healthy response to exercise rather than an injurious response. So I would suggest that when you're on the compound, you should be able to do whatever you like, and it should give you a positive benefit, right? If we're getting the dosing right and the compound's doing what we think it's doing. it should normalize the response to exercise. So it shouldn't necessarily matter whether you're doing oxidative or kind of like strength type exercises. I'm really excited to see what happens.
Joanne, this can be a two-part question for you, so we can get two at once. So we have a question about what's the criteria for future trials, and then specifically, are we planning to go into the non-ambulatory population?
Yeah. So we are in the phase 2 study in folks with Becker that we will be starting up. We will be enrolling ambulatory patients and who have a North Star within a certain range, like 10 to 32. We have a number of sites in the United States. We have not, until everything is finalized, we won't be sharing the whole list, but you will hear more on clinicaltrials.gov. Again, first half of the year, we hope to have a number of sites up and running. The site, the study, the open label study is just finishing enrollment now, but the phase 2 will be opening in the very near future, and can't speak to exactly where the centers are.
And we have some questions about timelines. And I don't know if you can really answer those, but of course, you're all waiting to know when this will be available. And Joanne, you can add to this, but all I'll say is we're working as diligently and as fast as we can to get this, to figure out what's going to happen with this drug. It's all a question still.
And hearing your questions and how makes us also appreciate how urgent the need is, and we've got a great group of people that are working incredibly hard to move these trials forward and hopefully get a therapy out to you.
If we have time, here's another question. Just what about reversal, Alan? Do you think that Edge5506 will reverse any damage already done?
So, great question, Matt. So you've got two different elements of the reversal question, right? So if you arrest injury, then that should allow the existing muscle fibers to become more healthy, right? So I'm not sure if that reversal is the right word, but certainly they should become more functional. Now, we've had some discussions about whether the fat incorporation in muscle is potentially alterable, because no one's really ever been able to arrest injury in muscle. That question has never been answered, but it's something that we're really interested in looking at. We'll certainly be doing MRI on limb muscles in the clinical studies, so we'll be able to measure it, right? Of course, I don't think you can replace fibrosis per se. Once that has really settled in and replaced muscle, now you can't really reverse that. But I'm hoping that even in a scenario where you have very little functional muscle left, you should be able to increase and improve the functionality of that muscle by stopping it breaking down.
Alan, another question for you. So what is Edge5506?
It is.
You might even want to ponder a little bit what a small molecule is compared to some of the others. And are there any allergy concerns?
No, not that we know of, allergy concerns. And we've done a whole bunch of safety testing, and this thing is is safe as far as we know it. Joanne described the side effects that we've seen in the healthy volunteer and Becker study, largely around dizziness and some sleepiness, but we've not seen anything else beyond that, really. Right now, of course, as you go into bigger studies, you never know what you're going to get, but so far, a pretty tolerable drug, right? What was the other question?
What is it?
What is it?
Where is it from, or how's it?
So we made it. So it's what they call a small molecule. So it's a drug much the same as something like Tylenol, right? So it's a small compound, much the same as any other medicine in a pill, right? We made it ourselves. We identified it by specifically looking for modulators of myosin and fast myosin at that. So we ran what they call biochemical screens using native muscle. So essentially mulched up muscle to look at modulators of myosin and then optimize this compound to have the best properties that it can, so that when we put it on muscle, we see that it decreases the injury response. The title of it is a selective fast myosin inhibitor, right? So it modulates that myosin molecule. A lot of big words there, sorry.
We can just reiterate this. So how does the compound work when you have partial dystrophin present versus full dystrophin present?
As I said on I don't know what it was, slide 5, we see it as a continuum, right? So at the very far end of the continuum, you have no dystrophin at all, right? So that when the muscle contracts, it gets stressed. A version of that happens if you have truncated dystrophin, right? It's still not fully functional, so that when muscle contracts, there's still a stress response. It may not be as big as the stress response that you get when you have no dystrophin at all, but it's still a stress response. And as such, we believe you can still alleviate that stress response. And certainly the biomarker data, the CK and the troponin data that we got in those first Becker folks suggests that that is indeed the case. All roads lead to stress, essentially, whether you have a truncated or an absent dystrophin.
Is it reversible? Like if you want to stop taking it and just you decide it's not right for you or whatever, talk about that a little bit.
Yeah. This is not an AAV gene therapy. This is a small molecule. You stop taking the medicine and it stops doing what it was doing, right? So much the same as medicine for a headache. If you stop taking the medicines for a headache, your headache will return, right? So in terms of ease of use, it doesn't get any easier, really.
I think we have gone through pretty much all the questions. So let's see.
Oh, there were some geographical questions about.
When are we going to know? When are we going to know the sites? And I think that I thought, Joanne, you want to answer that?
Yeah, and we will be posting it on clinicaltrials.gov. And also, you can sign up for our newsletter, and we will be updating there as well. And if you think of more questions afterwards, there is our email, and as you will get in touch with Abby. We want to hear from you. So thank you so much.
These are some great questions. Thank you.
So I think if I don't see any questions, that's going to be it for today. I'm so sorry again that I missed the beginning. So
okay.
Thanks all. Great. Thanks again.
Thank you.
Bye-bye. Bye-bye.